Hi everyone, I'm Brittany Nickham, a commercial product manager here at Beckman Coulter Life Sciences. Did you know that PCR cleanup hasn't changed since 1991?
No.
So we're here to bring you an update to that much needed PCR cleanup workflow. So we're going to introduce to you the Amnetic PCR cleanup system. It uses innovative magnetic technology to automate your bind, mix, and separate steps for your PCR cleanup. To show you this demo, we have Mindy here, an inexperienced user, to help me with the demo.
Yes, I'm Mindy Decker. I'm new to the marketing team and I'm new to the Amnetic too. And I'm really excited to learn how to use it with Brittany today.
Yeah, so Mindy, how inexperienced are you?
Really inexperienced. Like today I literally touched a pipette for the first time, so I'm a newbie.
Yep, so I hope this can show all of you how easy this is to use. Yeah. So let's go ahead and start. We turned on the Amnetic before we got started here. Did you get a chance to look at the user interface?
I did, and it seems really easy to use. And today we want to show everyone the PCR cleanup, and for that we just highlight that process and click the wheel, right?
Yep, let's go ahead and do that, yeah.
And then touch
click
it?
Yep. Yeah, so next we're going to go to step 1, which is initialize and add reagents. So let's go ahead and click that one too.
Okay, so it's saying we need a few things, and it's telling us exactly right here on the screen what we need. PCR samples, 75% ethanol, nuclease-free water, and Amnetic PCR sample reagents. It looks like we also have a multi-channel pipette and reservoir. Are those required?
Yeah, so the multi-channel pipette and reservoirs aren't required, and you could use a single channel pipette. But because this sets it up in a 24-well plate format, it's much easier to use a multi-channel and will help you get this done so much quicker.
Okay.
Yeah, and we also have all of our reagents set up over here.
Nice. And then I know we have this 8-well strip tube format. Can our customers use other sample tubes too for this?
Yeah, so this has our samples in it, and we're going to go ahead and add it. And yeah, so our customers can use single tubes, 8-well strips, or 24-well plates.
Okay.
So really easy to do what you've already doing for your PCR.
Nice. All right, so the interface says to vortex the Amnetic reagent for a full 30 seconds. Have we done that yet?
Yeah, so great catch. Before we started, we mixed up the Amnetic reagent. I'm just gonna mix it a little bit before we get started here, so the beads settle pretty quickly. So we'll go ahead and ready for that to be done.
I'll go ahead and add the bind reagent to our samples here. And this one?
Yep.
Is that enough?
Yeah, that's good.
How much do I add to our samples then?
So we're gonna add 90 microliters of our PCR cleanup reagent.
Okay.
I think it's set to 90, it's 090.
You're right, I think we did that before. So we're all set here.
Yep, so let's go ahead and add the reagent. So add tips. So for this time around, we have a 50 microliter. Let's go ahead and add. You'll go to that first press and add reagents. We'll add it to the samples. So for us, we're doing 50 microliters, but a customer could use down to 20 microliter samples, and they'll just add a 1.8x ratio of bind reagent. So now that you've added, we're going to go to step 2, which is the bind, mix, and separate step.
Okay. And then how does the Amnetic mix the samples with the reagent?
Yeah, so during this 3-minute period, the Amnetic 24 is creating magnetic fields using electromagnets. Within those fields, the super spray beads within the PCR cleanup reagent will mix with our sample. The Amnetic device will then create a different magnetic field to pull the beads to one side. But the other innovative part of this is really that super spray bead in the reagent kit. It was designed specifically to work with the device. There are no other beads on the market that should work with the device. The bead is really responsive to magnetic fields and therefore can mix and separate like we've designed it to.
Nice. So that means that it removes any pipette mixing from the cleanup process, and I know we've heard from a lot of customers that the pipette mixing can lead to a lot of fatigue and some guesswork, right?
Yeah, so actually I was having a conversation with former postdoc friend of mine. He has some issues with user variation in his lab, and he thinks that that could be due to user differences really within that pipette mixing. So the device really removes any guesswork and makes sure that user-to-user variation can be low in your lab.
Nice. Well, we're waiting for the 3-minute bind, mix, and separate step. Are there any steps that customers could use to change to optimize for their specific protocols?
Yeah, so I wanted to specifically point this out with you because in the genetic engineering, synthetic biology space, we know that customers really like to, make the workflow work for them. So you can run the bind, mix, and separate step multiple times. That's really helpful for really low or really high concentrations of DNA. Really make sure that your DNA is attached to those beads. We also have a Plasmid Prep kit, and running the bind mix steps multiple times is really helpful for large plasmids. Another step, which is like step 4 in the process, is ethanol dry. You can extend that time or shorten it. For this particular demo, we've shortened it for 30 seconds, and we'll come into that again. But if you have a really humid lab, you might want to run that dry step longer or shorten it. Then the last step we'll get to, which is the elution mix and separate step, you can run that multiple times as well. So again, if you have really high or low concentrations of DNA or large plasmids, it's helpful to run it multiple times. Also, if you walk off during that ethanol dry step and leave it for 15 minutes, you can run the elution step multiple times to really make sure that all of your DNA comes off the beads.
Nice.
So it looks like we have a little bit more time. You can see the progress bar there. Looks like about 15 more seconds.
I like that visual. It makes it really clear to see how much time you have left.
Yeah, it's really nice. I guess while we're waiting, maybe one of our questions we get from customers is, what types of samples can you run? And for this, for the PCR cleanup kit, you can run any PCR or enzymatic reactions. Say you have a restriction enzyme digestion or a Gibson assembly, you can use this particular kit for. And then we also have our Plasmid Prep kit, which you can use a bacterial overnight, and then you'll do the Plasmid Prep cleanup steps on the device. Okay, so it looks like it's almost done. Let's go ahead and get the pipette ready. We're going to change it to 200 microliters.
Sorry.
And that's 200. And we'll aspirate off all the liquid in here. And for our viewers, you can see that the progress bar turns green when it's ready for the next step. So it really gives you that visual of you're good to move on. Let's go ahead and add our tips here. And then we'll aspirate out the liquid. You can see that the beads are all the way to the side.
Oh, wow, the magnets do move the bead pellets to the side. Like one of our customers said, it looks really easy to aspirate.
Yeah, I think it will be for you, even as a brand new user.
Yeah,
so push it to the first stop, and then you'll go to the bottom, and you'll pull the liquid out. Yeah, so great job, Mindy.
Thanks.
And then you'll aspirate out the liquid into here and discard the tips. And we'll move on to our step 3, which is ethanol wash, which we're going to add 200 microliters of our ethanol.
This one here?
Yep. And then add it to the samples.
Like that?
Yep. And again, like that first step, all of the instructions are on the screen and tell you exactly what to do. And we'll wait 10 seconds, and let's go ahead and add new tips, and we'll take out the ethanol. Generally, you'll do 2 washes, but since this is just a demo, we're going to do 1. So let's go ahead and remove the ethanol. So we'll go to the first step, stop, and then the 2.
All the way down?
Yep. And then remove. All right, great job.
That didn't take much time at all. It looks like the ethanol dry step is next.
Yep, let's go ahead and go to the next menu.
So this is the last step before we have our clean DNA. I'll add 50 microliters of water for elution, right? After this is done?
Yeah, so we're going to wait 30 seconds. It's actually all in this here, but we have optimized this protocol for 3 minutes, but We're just doing 30 seconds for this particular demo. And then it also tells you how much elution to add. So we're going to add 50 microliters. It's what we found to be optimal, but you can go down to 20. So let's go ahead and change that to 050.
Okay,
yeah, great job.
Then add tips.
Yep. And they'll add water from the last trough on there. You'll add it to here because it already says green and done. So great job. And we'll go ahead and move to our elution, mix, and separate.
So while this step is finishing, can you kind of tell us what's going on here?
Yeah, so during this step, like in the bind, mix, and separate, the device is creating electromagnetic fields to mix your beads with the elution solution. So for here we have water. I think you can also use a couple other buffers, which are listed in the instructions for use. But during this step, the DNA is unbinding from the beads and mixing with your elution water.
Okay. Should we go ahead and answer some common questions that some of our customers have
Yes.
while this is finishing up?
Yeah, so we have like 30 more seconds left in the protocol. So one common question we have here is, how do you know what side the magnetic beads are on? Like, where do I put my pipette tip? So to help with that, we have etchings on the final instruments that show you exactly where to put your pipette tip and where the beads are. So you don't have to guess at where it is. It tells you exactly where it is. Kind of a theme throughout the whole procedure is you have instructions for exactly what you're supposed to do next. So I think it's almost done. Let's return to the main menu and let's go ahead and get those tips ready and your final sample tubes moved over and ready to take out that elution. We have clean DNA.
Well, thanks for explaining this to me. Hopefully I didn't butcher the process too much.
No, I think you're a certified scientist now.
Thanks for joining us.
Yeah, thank you.